Radiation hybrid mapping. Radiation hybrid mapping uses radiation such as X-rays to break the DNA into fragments. The amount of radiation can be adjusted to create smaller or larger fragments. These techniques overcome the limitation of genetic mapping and is not affected by increased or decreased recombination frequency. It is based on a statistical method to determine not only the distance between DNA markers, but also their order on the chromosomes. In RH mapping, human chromosomes are separated from one another and broken into several fragments using high doses of X-rays. Similar to the principle of mapping genes by linkage analysis based on recombination events, the farther apart two DNA markers are on a chromosome, the more likely a given dose of X-rays will break the chromosome between them and thus place the two markers on two different chromosomal fragments. The order of markers on a chromosome can be determined by estimating the frequency of breakage that in turn depends on the distance between the markers. This technique has been used to construct whole genome radiation hybrid maps. A rodent human somatic cell hybrid or artificial cells with both rodent and human genetic material which contains a single copy of the human chromosome of interest is X-radiated. This breaks the chromosome into several pieces which are subsequently integrated into the rodent chromosomes. In addition, the dosage of radiation is sufficient to kill the somatic cell hybrid or donor cells which are then rescued by fusing them with non-irradiated rodent recipient cells. The latter, however, lack an important enzyme and are also killed when grown in a specific medium. Therefore, the only cells that can survive the procedure are donor recipient hybrids that have acquired a rodent gene from the essential enzyme from the irradiated rodent human cell line. From these donor recipient hybrids, clones can be isolated and tested for the presence or absence of DNA markers on the human chromosome of interest, and the frequencies with which markers were retained in each clone can be calculated. This process is complicated by the fact that hybrids may contain more than one DNA fragment. For example, two markers retained in one hybrid may result from retention of the two markers on separate fragments or from no break between the markers. However, the frequency of breakage theta can be estimated using statistical methods and a load score, which is logarithm of the likelihood ratio for linkage, can be calculated to identify significantly linked marker pairs. Next, sequence targeted site or STS mapping. Sequence targeted site mapping resulted from DNA sequencing technology that allowed for the creation of detailed physical maps with distance measured in terms of the number of base pairs. At present, STS mapping is the most powerful physical mapping technique and the one that has been responsible for generation of the most detailed maps of large genome. A STS is simply a short DNA sequence generally between 100 and 500 base pair in length, which is easily recognizable and occurs only once in the chromosome or genome. To map a set of STS, a collection of overlapping DNA fragments from a single chromosome or from the entire genome is needed. In the example, a fragment collection has been prepared from a single chromosome with each point along the chromosome represented on average 5 times in the collection. The data obtained by determining which fragments contain which STS, this can be done by hybridization analysis, but PCR is generally used because it is quicker and has proven to be more amenable to automation. The chances of two STS being present on the same fragment depend on how close together they are in the genome. If they are very close, then there is a good chance that they will always be on the same fragment. If they are further apart, sometimes they will be on the same fragment and sometimes they will not. So, the data can therefore be used to calculate the distance between two markers in a manner analogous to the way in which map distances are determined by linkage analysis. In linkage analysis, a map distance is calculated from the frequency at which crossovers occurs between two markers. STS mapping is essentially the same, except that each map distance is based on the frequency at which break occur between two markers. To qualify as a sequence targeted site, a DNA sequence must satisfy two criteria. The first is that its sequence must be known so that a PCR assay can be set up to test for the presence or absence of this STS on different DNA fragments. The second is that the STS must have a unique location in the chromosome being studied or in the genome as a whole if the DNA fragment set covers the entire genome. If the STS sequence occurs at more than one position, then the mapping data will be ambiguous. Care must therefore be taken to ensure that STS do not include sequences found in repetitive DNA. The second component of an STS mapping is the collection of DNA fragments spanning the chromosome or genome being studied. This collection is sometimes called the mapping reagent, and at present there are two ways in which it can be assembled, as a clone library and as a panel of radiation hybrids. A radiation hybrid is a rodent cell that contains fragments of chromosomes from a second organism. The technology was first developed in the 1970s when it was discovered that exposure of human cells to X-ray doses of 3000 to 8000 rats causes the chromosome to break up randomly into fragments, larger X-ray doses producing smaller fragments. This treatment is lethal for the human cells, but the chromosome fragments can be propagated if the irradiated cells are subsequently fused with non-irradiated hamster or other rodent cells. Fusion is stimulated either chemically with polyethylene glycol or by exposure to Sendai virus. Not all of the hamster cells take up chromosome fragments. The routine selection process is to use a hamster cell line that is unable to make either thymidine kinase or hypoxanthine phosphoribosyltransferase, that is HPRT, deficiencies in either of these two enzymes being lethal when the cells are grown in a medium containing a mixture of hypoxanthine, aminopterin and thymidine, which is HAT medium. After fusion, the cells are placed in HAT medium. Those that grow are hybrid hamster cells that have acquired human DNA fragments that include genes for the human TK and HPRT enzymes, which are synthesized inside the hybrids, enabling these cells to grow in the selective medium. The treatment results in hybrid cells that contain a random selection of human DNA fragments inserted into the hamster chromosome. Typically, the fragments are 5 to 10 megabase in size, with each cell containing fragments equivalent to 15 to 35% of the human genome. The collection of cells is called a radiation hybrid panel and can be used as a mapping reagent in STS mapping, provided that the PCR assay used to identify the STS does not amplify the equivalent region of DNA from the hamster genome. A second type of radiation hybrid panel containing DNA from just one human chromosome can be constructed if the cell line that is irradiated is not a human one, but a second type of rodent hybrid. Cytogeneticists have developed a number of rodent cell lines in which a single human chromosome is stably propagated in the rodent nucleus. If a cell line of this type is irradiated and fused with hamster cells, then the hybrid hamster cells obtained after selection will contain either human or mouse chromosome fragments or a mixture of both. The ones containing human DNA can be identified by probing with a human specific genome wide repeat sequence, such as the short interspersed nuclear element, which is SINE, called LU, which has a copy number of just over 1 million and so occurs on average once every 4 kb in the human genome. Only cells containing human DNA will hybridize to LU probes, enabling the uninterested mouse hybrids to be discarded and STS mapping to be directed at the cells containing human chromosome fragments. The most common sources being expressed sequence tags, that is ESTs, Sslps and random genomic sequences. Expressed sequence tags or ESTs. ESTs are short sequences obtained by analysis of cDNA clones. Complementary DNA is prepared by converting an mRNA preparation into double-stranded DNA, because the mRNA in a cell is derived from protein coding genes, and cDNAs and the ESTs obtained from them represent the genes that were being expressed in the cell. From this, the mRNA was prepared. ESTs are looked upon as a rapid means of gaining access to the sequence of important genes, and they are valuable even if their sequences are incomplete. An EST can also be used as an STS, assuming that it comes from a unique gene and not from a member of a gene family in which all the genes have the same or very similar sequences. Sslps or symbol sequence length polymorphism. SSLPs can be used as STS in physical mapping. SSLPs that are polymorphic and have already been mapped by linkage analysis are particularly valuable as they provide a direct connection between the genetic and physical maps. Random genomic sequences. These sequences are obtained by sequencing random pieces of cloned genomic DNA or simply by downloading sequences that have been deposited in the databases.